On a beach south of Adelaide, scientists counted the bodies. 49 dead, 35 more dying where they lay. Greater crested terns, a bird that had survived ice ages, cyclones, predators, wiped out in days by something you cannot see, cannot smell, and until that week, Australia had never confirmed on its mainland. This wasn't an isolated tragedy, it was a confirmation. Because for years, Australia was the last continent on Earth, the last one, that this virus hadn't fully reached. And in the same month these terns died, Australian authorities confirmed something even more unsettling. The virus had jumped into a mammal, a long-nosed fur seal, its first mammalian case ever on Australian soil. This is H5N1. You've heard the name in headlines about egg prices and dead poultry. But as a microbiologist, I need you to understand what's actually happening beneath those headlines. Because right now, in dairy farms in the United States, in backyard flocks in India, in the wastewater of Bangladesh and Cambodia, this virus is doing something influenza viruses aren't supposed to do easily. It's learning mammals. By the end of this video, you will understand exactly how this virus infects a cell, why scientists are watching its mutations like a countdown clock, how doctors actually diagnose it in a lab, what treatment looks like today, and the one number, the case fatality number, that should make you take this seriously without panicking, because panic doesn't save lives. Understanding does. Let's go inside the outbreak dot. Let's start with where we actually are right now in 2026 now, not where the virus was 5 years ago. The global snapshot H5N1 first exploded globally back in 2020, and it never really stopped. By this year, it had reached wild birds on every single continent, including, as of this year, Antarctica itself, where it was confirmed on Heard and McDonald Islands. It has caused outbreaks across poultry industries worldwide. It has infected seals, foxes, cats, and, critically, dairy cattle in the United States, a species nobody expected this virus to colonize. Since 1997, when the first human infection was ever recorded in Hong Kong, the world has logged just over 1,000 confirmed human cases of H5N1. That number sounds small. Here's the number that isn't small dot. Historically, roughly half the people confirmed infected with H5N1 have died. Compare that to seasonal flu, where fatality is a fraction of 1%. This is why virologists don't sleep easy. Not because it spreads easily between people, because right now, it doesn't. It's because if it ever adapts to spread efficiently between humans while keeping anything close to that lethality, it would be catastrophic. The 2026 Hotspots Australia, as we opened with, went from 0 detections to over 200 confirmed wild bird events within weeks of the virus's arrival in 2026. Authorities have been explicit. This is likely to keep spreading. Conservationists are now racing to vaccinate iconic colonies, including the famous little penguins of Phillip Island, before the virus reaches them. The United States has the largest mammalian spillover event ever recorded for this virus. Since 2024, over 70 human cases have been confirmed, mostly farm workers exposed to infected dairy cattle or poultry, concentrated in states like California, Colorado, Texas, and Michigan. 2 of those cases were fatal, including the first US death, recorded in Louisiana. What makes the US situation unique in virology is this: influenza had never before established itself in cattle. It has now. That's a new ecological niche, and every new niche is a new opportunity for the virus to mutate. India has faced a brutal 2026 poultry season. Andhra Pradesh alone reported roughly 85,000 avian deaths, mass crow die-offs shut down parts of Chennai, and Karnataka had to cull thousands of birds at a state poultry farm near Bengaluru. India has also confirmed at least one human case this year, a child linked to a strain closely related to previously circulating Indian clades. Canada continues to report ongoing detections in domestic and wild birds, reinforcing what its food inspection agency has been careful to clarify. Avian influenza is not a foodborne risk. Properly cooked poultry and eggs remain safe. But the wild bird and poultry burden across Canadian provinces has been persistent and significant. The WHO and CDC position: here's what both the WHO and CDC agree on as of their most recent 2026 assessments. The risk to the general public remains low. There is still no confirmed sustained human-to-human transmission anywhere in the world. But both agencies use an identical word in every single briefing: And it's the word that should stick with you, .surveillance. Because low risk today is not a guarantee for tomorrow. Influenza viruses reassort. They swap genetic segments. And the more mammals this virus infects, the more chances it gets to roll those genetic dice in a body that's biologically closer to yours. Let's put this in perspective with a comparison, because numbers without context don't mean much. Seasonal influenza infects hundreds of millions of people every single year, and its fatality rate sits well under 0.5%. H5N1 has infected just over 1,000 confirmed people in nearly 3 decades, but historically kills roughly half of them. That's not a virus that's more contagious than seasonal flu. It's a virus that, when it does get into a human body, is devastatingly effective once it's there. Low frequency, catastrophic severity. That combination is exactly what pandemic preparedness planners lose sleep over. And here's the layer most headlines skip entirely. The case fatality rate isn't uniform across clades or countries. Cases linked to the newer US dairy cattle-associated genotype have generally presented more mildly. Often that conjunctivitis we'll get to in a moment, while cases in Cambodia and Bangladesh tied to different circulating clades have included the deaths we mentioned earlier. Same virus family, different genetic lineages, different clinical outcomes. That's not a coincidence. It's evolution happening in real time, branch by branch, and it's exactly why genomic surveillance isn't optional. It's the entire ballgame. So how does this virus actually pull off an infection at the molecular level? Let's go inside the cell. To understand why scientists are nervous, you need to understand what this virus actually looks like. Molecule by molecule, H5N1 belongs to influenza A, a segmented, single-stranded RNA virus. That segmentation is important. We'll come back to it. On its surface, it carries 2 key proteins that give it its name. H for hemagglutinin, the protein that lets the virus latch onto a host cell, like a key. And N for neuraminidase, the enzyme that lets new viral copies cut themselves free and escape the cell to infect others, like a knife. Here's the single most important concept in this entire video. Human cells in our upper airway, nose, throat, are lined predominantly with a receptor called alpha-2 6-linked sialic acid. Bird cells, especially in their gut, are lined with a different receptor, alpha-2 3-linked sialic acid. H5N1's hemagglutinin protein is currently built to grip that alpha-2 3 bird-type receptor far more efficiently. That mismatch is, right now, one of humanity's biggest protective barriers. It's why this virus struggles to spread person to person. Human infections mostly happen through massive, direct exposure, touching sick birds, inhaling aerosolized barn dust, contact with raw milk from infected cattle, not through casual contact like a cough on a bus. But here's the catch. Deep in the human lower respiratory tract, the alveoli, where oxygen exchange happens, we do have some of that bird-type alpha-2 3 receptor. That's exactly why, when H5N1 does infect a person, it tends to go deep, fast, causing severe viral pneumonia rather than a mild head cold. It bypasses the mild upper airway illness most flu strains cause and heads straight for the lungs. Now here's what keeps virologists up at night. Certain mutations, particularly one called PB2E627K, can improve how efficiently this virus replicates in the cooler temperatures of the human upper airway and in mammalian cells generally. We've already seen mammalian adapting mutations detected in cases tied to the US dairy cattle outbreak and in some severe human cases globally. Every new mammal this virus infects, a cow, a seal, a cat, a pig, is another roll of the dice for it to pick up mutations that make it more comfortable in a body like yours. That's why Australia's fur seal case and the US dairy cattle outbreak aren't just animal health stories, they're evolutionary pressure points. And remember that segmented genome I mentioned? If a cell, say a pig, which can catch both bird flu and human flu, gets co-infected by H5N1 and a human seasonal flu strain at the same time, those 2 viruses can literally swap gene segments. That process is called reassortment, and it's exactly the mechanism believed to have triggered past influenza pandemics, including 2009's H1N1. It's fast, it's unpredictable. And it's the scenario every surveillance program on Earth is built to catch early. This is why the US dairy cattle outbreak deserves far more attention than it got in most news cycles. Peer-reviewed analyses of the 2026 US outbreak describe something virologists had genuinely never documented before at this scale. H5N1 establishing sustained transmission within a mammalian livestock species, spreading cow to cow largely through contaminated milking equipment and shedding at extraordinarily high concentrations directly into milk. That's a brand new ecological pathway. Bird virus, mammalian gland, human exposure through an agricultural product most of us grew up trusting without a second thought. It's precisely why raw milk consumption is flagged so consistently in every public health advisory you'll see this year. Every infected cow is, biologically speaking, another incubator, another chance for the virus to fine-tune itself to mammalian body temperature, mammalian cell machinery, mammalian immune pressure. That's not fear-mongering. That's just how influenza evolution works, and it's exactly why the CDC and USDA have kept dairy herds under close genomic watch ever since. So if someone does get infected, how do doctors actually catch it? Let's step into the lab. Diagnosis is where clinical suspicion meets hard molecular science. Get this wrong, and you either miss a dangerous case or you trigger unnecessary panic. Testing isn't random. CDC and WHO guidance both center on exposure history first. Someone with flu-like symptoms and recent contact with sick poultry, dairy cattle, or a contaminated environment gets prioritized. And here's a detail most people don't know. In the current US outbreak, the predominant symptom in many confirmed cases hasn't been a cough. It's been conjunctivitis. Red, irritated, watery eyes. Clinicians have specifically been told, consider bird flu in a patient with pink eye and an exposure history, not just respiratory symptoms. Here's the actual diagnostic pipeline, step by step. That final step, sequencing, isn't just bureaucracy. It's how scientists caught that the Louisiana severe case belonged to a genetically distinct genotype from the strain circulating in dairy cattle. Sequencing is literally how we watch the virus's evolution in real time, clade by clade, mutation by mutation. And that distinction matters clinically. The genotype tied to dairy cattle exposure has generally tracked with the milder, conjunctivitis predominant presentation we mentioned earlier. The genotype linked to wild bird and backyard poultry exposure, the one behind that first severe US case, has tracked with more aggressive respiratory illness. Same headline virus, genuinely different clinical playbooks depending on genetic lineage. That's the entire reason a positive screening test is never the end of the diagnostic story. It's the beginning of a genomic one. Because of H5N1's severity, this isn't handled like a routine flu swab. Labs process suspected samples under enhanced biosafety precautions. And beyond individual diagnosis, health agencies run serosurveys, blood antibody testing on at-risk groups like veterinarians and farm workers, specifically to catch silent, mild, or asymptomatic infections that would otherwise go completely undetected. Because remember, if this virus is quietly infecting more people than we know, in mild forms, that changes the entire risk calculation. Now, the question everyone actually wants answered. If someone does test positive, what happens next? Treatment for H5N1 follows a clear, evidence-based protocol. And speed is everything. The frontline treatment is oseltamivir, brand name Tamiflu, a neuraminidase inhibitor. Remember that N protein, the knife that lets new virus copies escape an infected cell? Oseltamivir jams that knife. It doesn't stop initial infection, but it stops the virus from spreading efficiently from cell to cell inside the body. Both CDC and WHO guidance emphasize the same critical window. Antiviral treatment works best when started within 48 hours of symptom onset, which is exactly why exposure history and rapid diagnosis matter so much. In more severe or resistant cases, clinicians can turn to other antivirals like zanamivir, perimivir, or baloxavir marboxil, which works through a different mechanism, blocking the enzyme the virus needs to even begin replicating. Because H5N1 tends to strike deep in the lungs, severe cases can progress to viral pneumonia and acute respiratory distress syndrome. That means hospital management often mirrors the same intensive supportive care used for severe respiratory illness. Oxygen support, ventilatory support in critical cases, careful fluid management, and monitoring for secondary bacterial infection, which is a major driver of mortality in severe influenza cases, generally. On the vaccine front, there are two parallel efforts happening. For humans, candidate H5N1 vaccines exist and are held in national stockpiles for rapid deployment if the risk profile changes. But they are not part of routine seasonal vaccination because current human risk remains low. On the animal side, it's a different story. countries are actively vaccinating vulnerable wildlife populations. Australia, for instance, has been racing to vaccinate its most iconic and ecologically vulnerable colonies, including the little penguins of Phillip Island, before the virus arrives on their doorstep. Ultimately, though, prevention beats treatment every time. Here's what every major health authority, CDC, WHO, Australia's DAF, Canada's Food Inspection Agency, agrees on. Avoid contact with sick or dead birds and wild animals. Never drink raw or unpasteurized milk. Cook poultry and eggs thoroughly. Properly cooked food is not a transmission risk. Farm workers use PPE, gloves, eye protection, respiratory protection around sick animals. Report unusual bird die-offs to local agriculture or wildlife authorities immediately. That last one matters more than people realise. Remember, the Australian outbreak, the Indian crow die-offs, the US dairy spillover. Every single one of them was first flagged, not by a hospital, but by someone noticing dead animals and reporting it. Surveillance starts with ordinary people paying attention. So where does that leave us? H5N1 is not the pandemic. Not yet. Human-to-human transmission has not been confirmed anywhere on Earth. The general public risk, according to every major health agency, remains low. But this virus is doing something in 2026 that it has never done at this scale before. It's colonizing new mammals, from dairy cattle to fur seals, across more continents than ever before, including, now, Australia. And every one of those spillovers is a mutation opportunity. That beach south of Adelaide isn't just where this video started. It's a preview of what surveillance looks like in real time. Scientists, vets, and ordinary people, watching for the signal before it becomes a siren. As a microbiologist, my job isn't to scare you. It's to make sure that if this story escalates, and it might, you're not hearing about it for the first time from a panicked headline. You're hearing it from someone who explained the biology before it became breaking news. If this breakdown actually helped you understand H5N1 better than any headline could, that's exactly why this channel exists. I turn outbreak headlines into real microbiology, without the fear-mongering and without the oversimplification. So do 3 things right now, before you scroll away. 1. Hit subscribe and tap the bell, because when CDC or WHO updates this risk assessment, I will be covering it within 24 hours, not next week. 2. Drop a comment telling me which part surprised you most. The receptor biology, the conjunctivitis symptom, or the reassortment risk. I read every single one, and your questions shape my next video. 3. Share this with one person in your life who still thinks bird flu is just about egg prices. Because the biology deserves better than a headline. Public health isn't built by governments alone. It's built by people who understand why the rules exist, not just what they are. Stay curious, stay informed, and I'll see you in the next one.